In any metabolic pathway, we're going to have one or more enzymes that are going to increase the rate of a reaction by lowering the amount of energy it takes for that reaction to occur. And so this is what an enzyme is. An enzyme is a protein catalyst that binds to some substrate and either builds up or breaks down that substrate. So builds bonds between two molecules or breaks bonds and produces multiple products from one substrate. Some enzymes use cofactors or coenzymes to help. A coenzyme is an organic molecule. Typically, you're looking at vitamins, while cofactors are inorganic materials like zinc or copper elements. And their function is to assist the enzyme in modifying or breaking down, building up that substrate. Oh, okay, this is not working, so we're not going to watch this video. If you are interested in seeing this video, just click on, open up your internet browser, go to Google, your Google bar, and then just write How Enzymes Work, McGraw-Hill. Enter, and it will be one of the first things that pops up, okay? Enzymes can have different factors that will increase or decrease their reaction rate. Temperature is an example of something that will influence an enzymatic reaction. Because enzymes are proteins, temperatures can cause enzymes to speed up or slow down. But if the temperature gets too high, then enzymes will actually break down, because proteins can only work at certain temperatures. If the temperature gets too high, they will actually denature. The pH, and in the acidity or the alkalinity of the environment will influence an enzyme. If an enzyme is in too acidic or too alkaline an environment, they will break down again. Enzymes work best at wherever their optimum pH is. Not all enzymes work at the same pH or temperature. So some enzymes will work really well at, say, 37 degrees Celsius temperature. And some enzymes will work best at a neutral pH, but that doesn't mean all will. An example, pepsin is an enzyme that works in our stomach and works at acidic environments of 1 or 2 pH. And we have certain proteins that work very well at a much higher temperature. Think of your thermophilic bacteria. Enzymes can have regulators molecules that will either switch on or switch off those enzymes. So here's an allosteric region. And if a molecule binds into that allosteric region, it causes the shape of the active site to change. And so the enzyme substrate can no longer bind. This shuts down metabolic pathways, specifically when we don't need those pathways to run. And usually the allosteric molecule is the end product of the pathway. So if you have a lot of an end product, you don't need any more of that end product. Some of that end product will bind into the allosteric region and cause the enzyme to no longer function. When you need more of that end product, this molecule can unbind and be used. That also opens up the active site, and we can now produce more end product. Competitive inhibition is when a molecule binds to the active site. So in general, competitive inhibition is going to be seen with different poisons. Here's an example of a competitive inhibitor. Sulfa drugs act competitively to inhibit the pathway of producing certain molecules that bacteria need. So in this case, folic acid is not going to be able to be produced, and without folic acid, you can't produce certain proteins. And so here we have this PABA substrate that normally will bind, but when the sulfa drug binds, instead, it binds and the PABA can no longer bind. And so it blocks that pathway, and you're no longer producing the folic acid needed for the bacteria. Non-competitive inhibitors, usually when they bind, the action is reversible, but that doesn't always happen. There are certain non-competitive inhibitors that are irreversible and that basically make the enzyme no longer functional at all. And so some some chemicals are actual or poisons actually can bind to the allosteric region and cause the enzyme to no longer function. The whole function of this chapter is to talk about different metabolic pathways. And any metabolic pathway that runs is going to need ATP. ATP is the energy currency that our cells use. It's produced from a low energy molecule called ADP, adenosine diphosphate. When we add an extra phosphate to it, it becomes ATP. So there are three basic ways that we produce ATP. Substrate level phosphorylation, where you add a phosphate directly to the adenosine diphosphate, and you produce adenosine triphosphate. This gives us a small amount of energy, but it occurs relatively fast. Substrate level phosphorylation also is a very quick energy source, and all organisms can use this mechanism to produce energy. Oxidative phosphorylation uses an electron transport chain, and the electrons drive the pumping of protons, hydrogen ions, across a membrane, producing a proton gradient. And as those protons flow back from higher to lower concentration, they produce a lot of ATP energy. In photophosphorylation, we use sunlight energy to activate the movement of electrons and protons. Sunlight energy activates electrons that will then move into an electron transport chain. And as the electrons are shuffled from one energy level to the next energy level, down the energy gradient, protons are pumped across. These protons will eventually move back through the ATP synthase molecule or protein and produce energy. Photophosphorylation is used in your photosynthetic organisms. Oxidative phosphorylation is used in aerobic organisms. Substrate level phosphorylation is used in multiple organisms. One more term I like to talk about before we get into the different metabolic pathways, and this is oxidation and reduction. So prokaryotic organisms use a lot of different energy sources, and the electron acceptor, the terminal acceptor of electrons and hydrogens, will depend on the type of organism that is producing the energy. So if you're an aerobic organism, you're going to use oxygen as your terminal electron acceptor. But in anaerobic mechanisms, you might use something like an organic molecule, like pyruvate, or you might use a sulfur or nitrogen as your final electron acceptor. The term oxidation means loss of electrons, but in biology it also is associated with loss of hydrogen ions or protons. So here we have a reducing agent. He's going to lose his electrons and reduce another molecule. Here's an oxidizing agent. This one is going to become reduced and oxidize this one. So he gains he gains the electrons and he loses the electrons. He has been oxidized, he has been reduced. This reaction is known as a redox reaction or oxidation reduction reaction. We also have electron carriers that are associated with the production of energy. So we've talked about coenzymes, and I mentioned NAD and FAD, I know at least one time. NAD, nicotinamide adenine dinucleotide, FAD, flavin adenine dinucleotide. They are both electron carriers. Nicotinamide adenine dinucleotide phosphate, NADP, is also an electron carrier. When I think of these, I think of taxis. Their job is to pick up electrons and hydrogens anytime an enzyme breaks down a molecule and releases or oxidizes that molecule. NAD, FAD, NADP will pick up those electrons and those hydrogens and move them to wherever they need to go. That's their whole job. They're taxis, kind of like just moving from one stop to another stop and back and go back and go back and go back. And that's all they do all day long, okay? I will say that NADP is used in biosynthesis reactions, while NAD and FAD are used in catabolic reactions. And in any catabolic reaction, you're going to produce precursor metabolites. Precursor metabolites are just the intermediates in the catabolic reaction. So if you break a glucose molecule down, while it's being broken down, so if you start with glucose, you end with pyruvate, as an example. You're not going to just break glucose in half and produce pyruvate. You're going to produce a lot of different intermediate molecules before pyruvate is ever produced. And so down here you can see, like here you started with a glucose molecule. When it gets activated, becomes glucose 6-phosphate. If we don't want to break it down any further, or we don't want to modify it any further, we can just use that in a biosynthesis role, producing. Or here, down here, we have 3-phosphoglycerate. 3-phosphoglycerate can be used to produce amino acids. Pyruvate is the end product of glycolysis, but instead of moving into the citric acid cycle, we could take pyruvate and produce new amino acids. And so here we have E. coli. E. coli has a very diverse mechanism for producing energy. It can use anaerobic as well as aerobic means. If we grow E. coli in a glucose salt medium, it will be able to grow Glucose is the energy source that it will use, and glucose can be used to build up amino acids, nucleic acids, fats, and other carbohydrates. But we can also just break glucose completely down to produce energy. And so any of the molecules that enter, so as glucose enters, any of the molecules that have been modified as they're being modified can break off and move into a biosynthesis pathway instead of the catabolic pathway that it's on. Oh, sorry. So we're going to talk about some different metabolic pathways. Specifically, we're going to talk about glycolysis, citric acid or Krebs cycle, electron transport chain, fermentation, and the pentose phosphate pathway, which is part of photosynthesis. So here's aerobic respiration, which uses an electron transport chain to produce a lot of ATP via oxidative phosphorylation. Anaerobic respiration uses a different molecule other than oxygen as their terminal electron acceptor, but aerobic respiration uses oxygen as their terminal electron acceptor. We'll talk about fermentation for cells. A lot of cells can undergo fermentation. It's basically just that glycolytic pathway, and once you have no oxygen, or if you have no oxygen, then the glycolytic pathway, which produces a small amount of ATP gained, will convert the pyruvate back into some other molecule as it oxidizes the NAD. I'm going to stop here and we'll start on this next slide in our next video.